Institute of Biochemistry. So Irene hails from Italy, where she got her bachelor's in the Conservatory of Vicenza. She went on to get her master's in chemistry at the University of Padua, Italy, with a focus on organic chemistry and biochemistry. She received her PhD in chemistry from the University of Leipzig in Germany, with a focus on solid phase synthesis of peptides. Now, as a professor, since 2014, her research focuses largely on ligand-induced G protein coupled receptor signaling and other ligand-gated receptors, is a user of genetic code expansion, is a developer of genetic code expansion tools, and in a very short time, Irene has really contributed significantly to the field of GCE and GPCR signaling. So take it away, Irene.
Yeah, thank you, John. I hope you can hear me properly now. Yes, good. So I have tried, so I have only 20 minutes. I tried to put together a story, and this story starts, in fact, from my postdoc, which I did in the lab of Lei Wang, at that time at the Salk Institute. And everyone knows Lei, so he was a student by Peter Schultz. So he's one of the pioneers of the expanded genetic code technology, which I don't have to explain further now. That's good. For the first slide will be faster, as I promised. What we can give as a short overview, what we have hundreds of genetically encoded amino acids, and we know this, and I just picked a few just to give a few categories. So we have cross-linkers for photo and chemical, and photo and chemical cross-linkings. We have anchors for bioorthogonal chemistry. These are pretty much on fashion. Then we have photo-cage, photo-switchable amino acids, and biophysical probes. So there is quite among many others. So we have very much a wide chemical palette that we can pick our amino acids from. And I want to show you today some applications and a little bit of development involving photo and chemical cross-linking amino acids and anchors for bioorthogonal chemists. So let's start from the cross-linking. Cross-linking to study protein-protein interaction, to study protein-ligand interaction, to study whatever interactions as proteins. So which proteins? First, you have to pick a proteins. We have our PET proteins. These are G protein coupled receptors, just to make sure we are all on the same page. What are we talking about? We are talking of eukaryotic proteins that sit at the cell membrane, and they are activated by binding of a ligand, so-called agonist that activates the receptor. After activation, we have a productive interaction with the G protein. That's why they are called G protein coupled receptors. G protein dissociates, makes its signal. After this happens, GPCRs are phosphorylated by GPCR kinases, so they get phosphorylated. Once they are phosphorylated, they are still active, and they are bound by a protein called arrestin that arrests, in fact, stops G protein mediated signaling and triggers receptor internalization. And a second wave of signaling is still kind of controversial, but there should be a second wave of signaling that is arrestin mediated. So now I have started already in my postdoc using a genetically encoded cross-linkers to investigate the ligand receptor interaction. So this is old stuff that you can see in the literature. What I want to tell you about today is the application of genetically encoded cross-linker to study the receptor arrestin interaction, which is a more complex situation because we have two proteins. So this is a protein-protein interaction. You have barely no control about concentration of these proteins on the cell, very, very little. So with ligand is much easier because you add it to the cell, so you know how much you put and so on. But in this case, it's much more challenging. Why is this question interesting? Just because we have 800 receptors in other genomes and only 2 arrestins taking care of them, and it's not clear how the whole thing works. So it's clear that the system is highly flexible, and this feature is indeed what hampers structural characterization. So in 2022, we have only 6 GPCR-arrestin complexes that have been characterized mostly via cryo-EM. And we see a big variability within these complexes. Not only this, but we see also blind spots. So we see blind spots, which are the most flexible region that we cannot address. They elude the structural characterization. Also, you shouldn't forget that if you want to get a crystal of something, usually you need to do very heavy modifications so to get to be able to get the protein crystallized, or at least good enough for cryo-EM. So there are a lot of stabilizing mutations and nanobodies that are used. And so far, there are also only receptors of class A. Class A, so GPCRs are a big family, so there are several subfamilies. And class A is the bigger one, I would say the easiest one. They have very short N-terminals, so they are more compact. And so we don't have information about other classes with respect to structural information, with respect to arrestin binding. So what we did is that we picked a class B GPCRs, the parathyroid hormone receptor, and we explored the interaction with arrestin using genetically encoded cross-linker. Parathyroid receptor is a medical target, so there are already drugs out there for this receptor, and that's why the interaction with arrestin is interesting. Also because maybe we can get some hints to improve pharmacological profiles of drugs and so on. So that's a medical target, and we are trying to clear the interaction with the arrestin, because I suspected when we started the project in 2015 that nobody will be able to get a crystal structure or a cryo-EM structure before we are done, and I was right. So I will show you what we did. So the concept is that we have two steps. The first steps, you do a photo-crosslinking mapping. What does it mean? You take one of the two proteins you want to study the interaction of, and you scan the whole surface with a genetically encoded photo-crosslinker. So you know the incorporation is site-specific. So you can really map. You can use this photo-crosslinker as a proximity probe. The reaction is quite unspecific, so you capture whatever in the interacting protein. And then you need some assay, some readout for the occurrence of cross-linking. We had Western blot, spot at high molecular weight means cross-linking, no spot means no cross-linking. And we screened in this way the whole surface of the arrestin. So this is not a small protein. I mean, depends on the perspective, but we have about 400 amino acids. And to do this, we have established a protocol for high-throughput mutagenesis. And I can promise that you can get 100 mutants within 2 weeks. Yeah, it's possible. The bottleneck is the Western blot, so that you have to analyze. Then you have all this library of 400 arrestin mutant that carry BPA, so benzophenone crosslinker, at each position of the arrestin, and you start screening them. So we screened in batches of 24, and you see this is a Western blot targeted to the arrestin. So you see here the arrestin. This was the arrestin as an HAE to facilitate the detection, immunoblotting. And you see already in the N-terminals, so what was expected indeed of arrestin. We have this is the arrestin, and this is the cross-linking. You have cross-linking at some position. This is position specific. Some mutants don't express, but I can tell you we expressed about 95% of the mutants. So it's pretty good surface coverage. And then you make the next 24. Here we have barely cross-linking, and here we cover cross-linking in the third batch, and so on and so forth until you screen the whole protein. Once you are done, you get indeed the footprint of the receptor on the arrestin, because every black spot you get in Western blot is the covalent receptor arrestin complex. Once you plot on the surface of the arrestin all the positives, all the cross-linking hits, we say, so you really have like a footprint, so you know which positions of arrestin which positions of arrestin interact of the receptors. And what we could see is, you see here is different perspective of the arrestin, that the most largest interaction surface is this end domain here, and so on. So this is nice, but it's still not enough, because in fact you want to know which position. So in this case, with the photo-crosslinker, you know the position where you incorporated the photo-crosslinker. What you don't know what you have captured. Mass spec people will promise you we find it out. You can go with that if you want to be a bit faster and screen much more combinations. Then you can go with the pairwise cross-linking with no mass spec. And the concept is that you have a proximity-induced reaction. So it means you have an electrophile unnatural amino acid that reacts specifically with the nucleophile canonical amino acid. So you don't have to genetically encode the nucleophile. This is already easy as canonical amino acid in your protein. And you screen the one protein with the unnatural amino acid with the electrophile, and the second protein with the nucleophile. So then you ask me, why do you did the photo cross-linking mapping? Poor PhD student. Yeah, we did it because in this case, you don't have, at this point, you don't have to screen anymore 400 arrestin mutants, but you just pick the ones, the subset of ones that gave photo cross-linking. You pick basically only the good photo cross-linking ones, and because it's a combinatorial, then you want to reduce as much as possible the lines of your matrix. So you want as little as possible positions so that your matrix will still be manageable in a reasonable time. So you have on one side the non-canonical amino acids, so the electrophile. You don't need to clone anything. You can keep the library you use for photo cross-linking, just change the synthetase. And then we screened the intracellular side of the GPCR with cysteine, because this unnatural amino acid will use the target cysteine. And then every pair you get is like playing Battleship, right? Every pair you get here gives you a pair of amino acids that are proximal in the complex. In first place, we have screened we have worked even with the wild type receptor, because it has 4 cysteines in the intracellular domain. So this is fantastic, because you have really the native receptor. So we didn't do anything, no mutation, nothing. The receptor is the way it is as nature made it. And we have our arrestin with the electrophile on the receptor. And you see, we find cross-linking so we tested 24 positions covering the footprints, and then we check this position. This is the position where the electrophile in arrestin is, and this is the interaction with the wild type receptor. So we found several cross-linking, and what we did is that we replaced one by one this cysteine in the receptor with serine. Serine does not react with this nucleophile, with this electrophile. So every time by putting a serine, you lose the cross-linking. It means you discover which cysteine is responsible for this cross-linking. And so already from this data, you can already infer more or less the orientation of the whole complex. And then we went on and screened the whole intracellular phase with these cross-linkers, with the cysteine, pardon, and we get a matrix of interaction. We did everything in triplicate, do statistic analysis, and after all this, we left with just 136 statistically validated proximity point. I'd say just because we had more than 200. So it means we could even get more out of our data by examining dynamics. But the good cross-linkings were 136. So what do you do with this treasure? So it means you know 136 pairs of amino acids that are close to each other. So then you have to go to molecular modeling. So we did molecular modeling on the PTH receptor arrestin complex, and we put constraints. So we constrained, we told the computer that it should build a model where the hits of our pairwise cross-linking. So it means these pairs of proximal amino acids, they should not be far more far away than 10.2 angstrom, which is the distance of the thioether that's generated by the electrophile cysteine reaction. So in this case, what we got is a beautiful model of the arrestin PTH interaction. Of course, you need here some starting points. So you need either an homology model, or like in this case, we had there is some structure of a PTH with G proteins. So we used existing partial structures as starting point, and then we made our model. And the only thing, I will not tell you details because it's not important in this context, but you see all the colored parts are the parts where we have constraints. So what I want to say with this is that we can really follow very, very detailed all the interaction parts in this interface. And in this case, we have a lot of information also about parts that are not resolved in any of the existing complex. So, and another little thing I want to give you, if you want to do this kind of experiment, is that it can happen that you have some points where you have your electrophile that keeps a lot of cross-linking, a lot impossible, like 12 cross-linking. So how can you possibly put 12 amino acids within 10 angstrom? That's impossible. But so you do, of course, an ensemble of models. But what we did is that when you do molecular dynamic simulations, you see that all pairs come within the distance of cross-linking within the time of simulation. So this is a glance into the dynamic of this complex. So more you will hopefully hopefully read in the literature soon. We are submitting the revision, so I hope it's a matter of weeks. And another thing I want to present to you today is a different story. So the incorporation of unnatural amino acid to label proteins, so bioorthogonal chemistry. And you know better than me that all this is last generation amino acids, right? These are all new amino acids that have been genetically encoded using the pyrolysin synthetase. And this is very cool, everyone knows. But what people do not emphasize in papers is that often this the pyrolysin system gives not such a amazing expression in mammalian cells. So we struggled at the beginning with the existing system with the low incorporation rates in mammalian cells, and this is for challenging membrane proteins deleterious situations. PhD student was hating me. And so we were thinking, so why is it so bad? It's so good in bacteria. Why is it so bad? And so we focus our attention on the tRNA. This is very special guy because first, you know, it is the anticodon stem is longer than canonical RNA. There is one base missing at the hinge between acceptor stem and D loop. And this stem, D loop is very, very short. And the usual motif tip CC is missing. The variable loop is almost not existing. So the point is, if you work in mammalian cells, the expression you need to express to transcribe, not only to transcribe this tRNA, but you also need to process it. And so we suspected that this very strange tRNA is the reason why mammalian cells don't like this system so much. So we look for a compromise where we still had good recognition by the synthetase, but still orthogonality and compatibility with endogenous components. And what we did is that first we have observed that there is another tRNA in mammalian cells that looks exactly like the pyrolysyl tRNA, and this is the mitochondrial tRNA, the mitochondrial serine tRNA. And what we did, we took the scaffold of mitochondrial serine tRNA and we imported imported recognition elements of the pyrolyzed tRNA. Something similar had been done before in bacteria. I don't want to lie. I suspect this was the Ditazole, I assume. I should double check, so I apologize if I made a mistake now. Other thing is that we try to import human conserved motifs of canonical tRNA into the pyrolyzed tRNA. We have an assay to check our tRNAs. This is also published, and you see this is it's a fluorescent-based assay. So we read we have a GFP readout basically, and an mCherry control. So this is the efficiency of the normal tRNA. So the pyrolyzed tRNA, and you see that some of our designer tRNA are dead, but there are a few others and some others that are really good. They are much better. So we put this one in a plasmid system with the 4x tRNA expression cassettes. We have always the U6 promoter in these cassettes and some T-rich trailer. And you see here, so I apologize for this shade of gray blot, but in fact you see that the first bar is always the pyrolyzed tRNA, and the second bar is the one that comes from the mutant series where we have imported mammalian motif into the tRNA, and this one is the chimeras with the serial mitochondrial tRNA. So you see that basically we have doubled the incorporation efficiency for BOC and even for CPK, PRK, lysine set. So same. So we have much better expression. So you can ask, why is this expression much better? So then comes the analysis, northern blot. So what you can see here, this is the regular tRNA, and this is our best tRNA. This is M15, mutant 15 tRNA. And you see that this is a control, expression control. This is in vitro expressed that we use as a reference for concentration. This is identical in the two blots. And you see that the normal tRNA gives a very little expression. And even if you use multiple cassettes, which is a very much used thing there are also construct with H cassettes and system with 8 cassettes and whatever. And what we want to show you here, I hope I can convince you that is not the case of having more cassettes, but the case of having a better tRNA. Only one copy of our tRNA gives more tRNA as 4 copies of the bad tRNA. So this is the acylated tRNA, this is the non-acylated tRNA. So you see, we have improved stability in the cells, and the one reason can be that in our mutant tRNA we have replaced this hinge between the two stems with the canonical hinge GC, and instead of AU, and we have several stabilizing mutation, including, and we have also done pseudouridine analysis, and we found that we have more pseudouridine, one more pseudouridine in the mutant tRNA, and this is a position that is known to increase stability. And so now is the big proof. So can we improve GPCR incorporation of non-canonical amino acids in GPCRs? Answer is yes. So you see, this is regular tRNA. This is our tRNA. Regular tRNA, our tRNA. Regular tRNA, oh God, there's nothing here. Our tRNA, we can still save some expression. So, and also we can use this system to incorporate 2 amino acids, but everything here is published. So if you are interested, you can check on it. And what we did as application is that we use then the system to label GPCRs on the surface on the cell, and not just on the surface on the cell, but you see this loop, this little loop here. So there is no other way to label this loop as using a genetically encoded expansion, genetic code expansion, because if you insert fluorescent protein here, your receptor will be dead. This is very sensitive region. And we can label this region only thanks to this very, very minimal mutation. So you see these are 3 different receptors. And the important part is here, you see these points here are the receptor that internalizes, goes inside the cell after you stimulate it. It means it's healthy, it's still working. And I bet there is no other way of doing this with the other system. We also demonstrated that where each of the receptors we have investigated, we could find at least one position give you quantitative yield, which is important if you have to do stoichiometric analysis, I don't know, single molecule tracking, dimerization, so you don't want to have dark receptors around, so that's important. And we also show that these receptors are faster compared to receptors tagged with GFP. So there is the diffusion coefficient is more natural. So unnatural amino acids are simply beautiful. Next thing is that this is a work in progress. You know, fluorophores are environmentally sensitive. If somebody tells you a fluorophore is not, it's probably lying, or it didn't do the right experiment. If you incorporate this, you label receptors at some positions, and you stimulate the receptor. You see here you have the fluorescence. You stimulate the receptor, bam, the fluorescence changes. You wash it out, recovery. You add another activator, fluorescent change, and so on. So in this case, you can monitor the dynamics of GPCR activation. So we have a work, this is a collaboration with Andreas Bock here in Leipzig, and we have a work in progress on this, and this is not a matter of weeks, but I think a matter of months. I hope you will see something in the literature about this. It's really, really exciting. Okay, so GCE for all was the title. So all plasmids that we use and are not presented by others already, we have deposited in AdGene. They are all humanized for mammalian expression, and we have here our best tRNA, for instance. Here is the cross-linkers, azide, BPA, and this is the chemical cross-linkers. So with this, you can map protein-ligand interaction, protein-protein interaction. You can label proteins, and in this case, you can also, through fluorescent changes, investigate dynamics, but also dynamics of dimerization or so. And to these projects, our collaborators, so my group is small, you see, I mean, some people have changed, but the size didn't really change. And so to this project started with my postdoc by Lei Wang, and then we have collaborated for a RESTIN project with Seva Katric and Seva Gurevich from LA and Vanderbilt. Mario Merl is an RNA specialist, helped us with the tRNA project. Annibale, Paolo, Andreas, and Martin Lose are the imaging guys that helped us with the labeling project. And these are all nice people who gave us money. Basically is the German Research Foundation that financed us most. And with this, 25 minutes, 38 seconds.
Great job. Thank you so much for the great talk.
I made it. I'm ready to take questions.
So there's 2 questions. I can read them to you. You can answer. Just have you considered using the photo-crosslinker Uaa with the transferable MS label to identify PPI?
Yeah. Yes, we did. And in fact, I will tell you that we use the photo-crosslinker without transferable MS label. And in some positions, we could identify the. This is also published. We are not first authors, so this is a collaboration with Jean-Pierre Villadaga in Pittsburgh, and they could find identify in the receptor using mass specs. Is very simple, just regular IP. The cross-linked part in the receptor, but our experience says that. This is really hard. If you want to go for large-scale mapping of surfaces, you are better off if you have a high-throughput assay to test more combinations in smaller time. We are working on this. Otherwise, I get no PhD students anymore. Too many Western blots. So that's important. Yeah, we don't exclude to go on with mass spec. On the contrary, we are also working on this. But so far, I have to say it's still challenging. We have so little protein that we get of these samples. It's really tough.
Great. So the next question, is there any reason why you pick M-Barkeri pile-RS for incorporation of TCO rather taking the better incorporating M-Mazai pile-RS?
Oh, I am so surprised because actually in our hands, we have done a lot of experiments with both. Actually, at the beginning, my student was not sure which one we should take. And so he was studying both. And I have to say, we didn't find a big difference. What was a big improvement for us when we decided to humanize the genes of both. And with humanized genes, I have to say that he didn't see big difference. At the end, we stuck with the Barkeri because it's a little bit smaller, and I don't know, it was more nice to us. Just there is no really a deep reason for that. So they were equivalent in our hands, and so what we have seen that might help is the M. albus. This is something that we did later, so it's not published yet. But I don't know if my students is there, but it was TCO, I believe. Yeah, it must have been TCO. And she tried M. albus with the M. albus tRNA, not with the one that is orthogonal to the other pyrolysis. So it's not the one that is used when you want to incorporate 2 amino acids. So really the natural M. albus tRNA, this was a bit better than MB. But we didn't see this improvement with MM. Maybe we check again, but yeah.
And then a question from the same person, is M15 compatible with M-Mazai pile-RS?
Yeah, absolutely. And it is also, I don't know. I don't know if we did the experiment already. If it goes also with M. albus, I cannot tell you. But definitely it is compatible with both.